Hello, everybody. Thanks for Laboratories to invite me to present this work on SARS-CoV-2. My name is Ricardo Rajsbaum. I am an associate professor in the Department of Medicine at Rutgers, New Jersey Medical School, and the Center for Virus-Host-Innate Immunity at Rutgers. I'm excited to join these events and tell you a little bit of the work that we have been doing on COVID. Our lab is interested in studying different aspects of host-virus interactions, especially the role of the innate immune response during different virus infections. During virus infection, the host cell recognizes viral infection via pattern recognition receptors that are expressed in the cytoplasm or in endosomes, like toll-like receptors or Riga-like receptors. And this viral recognition triggers multiple signaling pathways that culminate in the production of pro-inflammatory cytokines and type 1 interferons. We are particularly interested in studying type 1 interferons because of their well-recognized role as antiviral cytokines. Interferons are then recognized by their receptor in other cells in autocrinoparachal manner and trigger their own signaling pathways to induce interferon-stimulated genes or ISGs, which are actually the proteins that inhibit or have effector functions against viral infection. Of course, this is not perfect. The viruses have evolved to express antagonists or proteins that can help the virus escape or antagonize this response. We have been studying all of these aspects of the innate immune response on different virus infections like Nipah, Ebola, influenza, and Zika. And during the pandemic, we got interested on trying to elucidate the mechanisms by which SARS-CoV-2 might evade the immune response or be recognized by pattern recognition receptors. During these couple of years, during the pandemic, has been very clear that the cells, when they are infected with SARS-CoV-2, can sense the infection via pattern recognition receptors. And there have been a few studies, and there is debate whether the RNA pattern recognition receptors like RIG-I or MDA5, which one plays a more important role during recognition of viral infection. A few studies have highlighted the role of MDA5, but others have also suggested that RIG-I may play also a role. And upon recognition, as with other virus infections, SARS can also trigger this signaling an immune response, producing large amount of pro-inflammatory cytokines and chemokines. And in humans, it has been shown that actually a lot of these pro-inflammatory cytokines are responsible for damaging tissue or for this hyperinflammatory response that can cause disease. And these cytokines can also cause cell death. Obviously, type 1 interferons can also have an antiviral effect, but can also act as inflammatory mediators. So one of the biggest questions is how SARS-CoV-2 have also developed these different mechanisms to antagonize this, how this might affect not only the inflammatory response, but also blocking, again, interferon and allowing the virus to recognition. So this is one of the subjects that we have been studying, and this work actually was led by Peiyong Shi and Hongji Xie at UTMB, and we collaborated with them to elucidate the role of SARS-CoV-2 proteins in antagonism of the immune response. And this amazing work, the group of Peiyong cloned basically all the proteins of SARS-CoV-2 in expression to be able to study their independent roles during virus infection and interferon production. For these studies, what we use is a well-established reporter assay in which you can overexpress the two CAR domains constitutively active, and basically what it does is mimics recognition of viral infection. Is like if RIGI would be recognizing RNA present in the cytoplasm, but there is no virus here. It's just the two CAR domain of RIGI that is constitutively active and triggers this response via the transcription factor IRF3 that can activate this interferon beta promoter lucifer assay. So then with the presence of the CAR domain that is activating the pathway, now we can also overexpress each one of these 27 different proteins of SARS-CoV-2 to study their independent role as interferon antagonists. And as you can see here, there were 4 proteins that clearly gave an effect when they were overexpressed in this assay. So what you can see is that when you overexpress RIGI, there is interferon induction. The majority of the SARS-CoV-2 problems, at least in these conditions, did not inhibit the pathway, but in NSP1, NSP6, NSP13, and ORF6 showed a really strong antagonism or inhibition of the interferon induction with this assay. So NSP1 was already known to have antagonism effects, including with SARS-1. So in these studies, we focus on the other proteins. And so what you can do to figure out, to narrow down the mechanisms of inhibition is you can overexpress each of the proteins that are downstream of RIGI in this pathway to mimic activation of the pathway at different steps, so you can study which of these steps are targeted by the viral proteins. So for example, you can overexpress MAVs and you will get an interferon induction, this luciferase reported assay. So what you can see here, and there is a lot of data, so I will try to guide you through this data. And basically what you can see is that if you overexpress MAVs, you will get induction of the interferon reporter, and all of these proteins, basically NSP1, NSP6, NSP13, and ORF6, they all can inhibit the pathway when MAVs is overexpressed, which means that the inhibition is occurring either at the level of MAVs or downstream. So you can continue doing this with downstream known signaling factors. So if you overexpress IKK epsilon, again, you can have induction of the pathway, and you can see that all of these proteins can still inhibit this response, suggesting that, again, that these proteins are acting downstream of IKK epsilon. The same thing you can do with TBK1, and you get similar effects. But what it's interesting is when you get to IRF3. If you overexpress IRF3, you can see that ORF6 can still inhibit this pathway, but NSP13, NSP6 lose this antagonism. So basically what this means is that NSP6 and NSP13 might be acting upstream of IRF3 and ORF6 downstream or on IRF3. So this can be corroborated by looking at phosphorylation of IRF3. And so what you can see here is that indeed when you stimulate the pathway with a double-stranded RNA or poly-IC, poly-IC is a mimic of double-stranded RNAs, but it will trigger the pathway. You can see that indeed NSP6 and NSP13 can inhibit phosphorylation of IRF3, but ORF6 does not. So this means that ORF6 is probably, well, you can also corroborate this by looking at phosphorylation of TBK1. This is also very interesting because you can see that as while NSP13 inhibits phospho-TBK1, NSP6 does not. So basically what this suggests is that NSP13 and NSP6 are acting at the level of TBK1 or between TBK1 and IRF3, but ORF6 is really acting downstream of IRF3 because it does not inhibit phosphorylation of IRF3. We could follow up and demonstrate that one of these proteins can interact with TBK1, and basically still studying the detailed molecular mechanisms of how that inhibition occurs. But looking at ORF6, what you can see really is that when you overexpress poly-IC, IRF3 is translocated to the nucleus as normally it occurs, but you can see that when in the presence of ORF6, translocation does not occur efficiently, suggesting that indeed, corroborating that ORF6 is inhibiting translocation of IRF3 to the nucleus, so downstream of IRF3 phosphorylation. And to look a little bit more into the mechanism, we could find that there's an interaction between ORF6 and these proteins, KP and A, which are part of the nuclear pore that are important for translocation of proteins to the nucleus. So this provides some basics for mechanistic insights on how the different proteins of SARS-CoV-2 might be inhibiting interferon production pathways. I'm not going to go through the data, but in these studies, we also performed a similar interferon reporter assay, now looking at the interferon signaling pathway. So basically using interferon stimulated gene promoter that is sensitive to treatment with interferon. And now you can do overexpression of all of these proteins to look at their potential activity as antagonists of the interferon signaling pathway. And there were even more proteins from SARS-CoV-2 that were detected to have activity, indicating that really SARS has many ways to antagonize the interferon response. Even though it appears from other studies that interferon, that SARS-1 might be that SARS-2 might be less sensitive to the effects of interferon than SARS-1. To follow up, we are also interested on looking at how different viruses and how the immune response is regulated, how the signaling pathways are regulated. In this case, we are really interested in looking at post-translational modifications like phosphorylation, and including ubiquitination, which we know can regulate activity of many proteins. So, for example, we know very well that there are different types of polyubiquitin chains. So ubiquitin is a small protein that can form polyubiquitin chains depending on what lysines are modified in ubiquitin, and can form these polyubiquitin chains with different functions. Why is it so important? I mean, ubiquitin is a small protein and can covalently modify other proteins via its C-terminal glycine-glycine residue, that it's important for modifying a lysine residue in another protein. And as I mentioned, because ubiquitin has 7 lysines, each of which can be modified by another ubiquitin to form a polyubiquitin chain. These chains have their function really because of the shape that they form, or this topology that can form depending on where the linkage is formed. And this is due because other proteins that will be activated or will have a function depending on ubiquitin rely on ubiquitin binding domains that will have to interact with these polyubiquitin chains depending on their shape or topology. So you can think about these like a key and a lock, in which this type or shape of polyubiquitin chains have to match the domain. going back to the studies that I show you on the different proteins of SARS, we looked at more detail the mechanism of ORF7, ORF7A inhibition of interferon signaling, where it was really interesting. Also in collaboration with Pei-Yong Shi lab, they found that this ORF7A it's heavily ubiquitinated. And they could actually identify, mutating each one of the lysines that are contained on ORF7, that this lysine 119 it's responsible for ubiquitination. This mutation, when you mutate ORF7, you can see there is a reduction of ubiquitination on this site. What is interesting is that this ubiquitination is actually important for full interferon antagonistic function of ORF7. So what you can see here is that using that luciferase reporter assay that I mentioned, is that when you utilize this mutant K119 of ORF7A, you partially lose the ability to inhibit the interferon signaling pathway. And to look a little bit more in detail, we can see that ORF7A can inhibit phosphorylation of STAT2, but not phosphorylation of STAT1. And when you use this mutant of ORF7, you lose that ability to inhibit phosphorylation of STAT2. You can look at this also at the level of translocation of the ISGF3 complex to the nucleus. So basically, phosphorylation of STAT2 is important for the formation of this complex. So if it doesn't phosphorylate well, you will have a deficient translocation to the nucleus. And this is what you can see here, is that when you stimulate the cells with poly-IC, here is interferon stimulation, you will see that there is presence of STAT1 in the nucleus because it's part of the ISGF3 complex, but in the presence of the wild-type ORF7A, you have less translocation to the nucleus, but you lose this effect when you use the K119 mutant. So now I want to switch to another interesting part of the innate immune pathway that we have been studying, and it's trying to figure out or elucidate new factors that may be important in the recognition of different viruses, in this case SARS-CoV-2. So we have been trying to establish novel approaches to identify novel pattern recognition receptors that would be involved in these pathways. So for this, I want to go back to some studies that we previously studied to explain one of these novel approaches that we have developed. So in 2014, we published that this E3 ubiquitin ligase, TRIM6, so is one of the enzymes that is required for the formation of polyubiquitin chains. We found that this E3 ligase can form an atypical form, or not very common form, or not very well studied form of ubiquitin chains. TRIM6 can generate ubiquitin chains that are linked via the K48 lysine of ubiquitin, which usually is known or is studied for its role in degradation of proteins by the proteasome. But what we found here is that TRIM6 can make these chains that are not covalently attached to any protein. So they have a free glycine residue in the end of the chain and are not covalently attached to any protein. And basically what they do is that they can promote protein-protein interactions. And in this case, it's IKK epsilon, the kinase that is involved in interferon production and signaling. And TRIM6, by generating these polyubiquitin chains, can promote the oligomerization of IKK epsilon and autophosphorylation that is important for its activation to phosphorylate downstream substrates, like, for example, phospho-IRF3 or STAT1. What we have been trying to do since then is to figure out if we can use these to an approach, or design an approach in which we can isolate these polyubiquitin chains from the cell and study which proteins would be interacting and identifying roles of these proteins that would be interacting with non-anchor ubiquitin. And basically, one of the reasons to study this in terms of recognition of viruses is because in 2010, it was also reported that the pattern recognition receptor RIG-I could also interact with different types of polyubiquitin chains. These are K63 linkage, but they were also non-covalently attached, or basically a similar type of mechanism to the one that we show in the case of IKK epsilon and these K48 chains. So this implicates activation of pattern recognition receptor partially by unanchored polyubiquitin chains. So what we try to establish is an approach to isolate these chains from cells coupled with mass spectrometry. So basically what we did is to use chains, ubiquitin chains that are purified and basically they are unanchored because they're not covalently attached to any protein. They are just polyubiquitin chains. This can be K48 or K63 linkage, but they have a free glycine residue, so they are not attached to any protein. You can mix lysates from cells and let them proteins attached to these chains and isolate these chains and analyze proteins that will be interacting by mass spectrometry. This was work done by a former graduate student in my lab when we were at UTMB, Adam Hayes, and basically what we found here is a protein that is called DHX16. It's an helicase, an RNA helicase that was previously known to act as a splicing factor. It was not known to have any function in the interferon response, but it was known to act as a splicing factor. So we were very interested about this because of its potential role as interacting with RNA and how this could be potentially involved in any of these pathways. So the first thing what we did was to corroborate that DHX16 can interact with an anchor polyubiquitin chain. So in this assay, we can overexpress DHX16 in the presence of HA ubiquitin. We immunoprecipitate HA ubiquitin, so we isolate ubiquitin. And to demonstrate that the interaction is with non-covalently attached DHX16, we can wash with a high stringency buffer containing high salt that would only break or destroy non-covalent interactions. And what you can see here indeed is that you lose interaction when you wash the beads containing DHX16 and ubiquitin. Another way to demonstrate this is in vitro by using semi-purified DHX16 and add in the addition of purified unanchored free ubiquitin chains, and you can see that we indeed can get interaction of ubiquitin chains of the K48 linkage type, but not K63 linkage type. So we further wanted to know the effects on the interferon response. So we did, well, in general, like we did a knockdown of DHX16, which was very efficient, and we performed next generation sequencing to see what factors are downregulated upon in downregulation in a knockdown of DHX16, and we were really surprised to see that this knockdown was really biased toward genes that are related to the interferon pathway. We were expecting to see a broader response because of its previous role in splicing. But we found a lot of ISGs, interferon-stimulated genes, that were downregulated when a knockdown was performed on the DHX16, and this implicates the DHX16 in the interferon response. So we performed a lot of more experiments to confirm these assays, but what we did next was to use knockdown again and infect with different viruses to see whether indeed DHX16 has an effect or a function upon virus infection. We use different viruses like influenza and Zika, in which we could see that there is a reduced interferon induction when you infect with these cells in knockdown DHX16. We could also see the same thing when we infect with SARS-CoV-2, and indeed SARS-CoV-2 had increased viral titers also when knockdown of DHX16, most likely because of the defect in interferon induction. But this was also interesting because not every virus that we tested was affected by DHX16 knockdown, for example, Sendai virus, suggesting that DHX16 has a specific role towards different viruses. So to further try to narrow down which type of RNA is the one that is recognizing or acting with DHX16, we use different RNAs to stimulate the cell, and we use here a 5' triphosphate RNA that is known to stimulate very specifically the RIG-I pathway via recognition by RIG-I. And you can see that knockdown of DHX16 has a huge effect in induction of interferon when it's stimulated by this type of RNA, suggesting that RIG-I is involved. We can further demonstrate that there is DHX16 couples with RIG-I, because if we overexpress DHX16 alone, we can get some interferon induction, but knockout cells, RIG-I knockout cells, fail to induce any interferon when DHX16 is overexpressed. So really indicating that DHX16 requires RIG-I for this interferon induction. We further wanted to go deeper into the mechanism and how DHX16 may be coupled with RIG-I for RNA recognition. So if you use a RNase enzyme to cleave RNA, you can see that first, when you cleave RNA, the interaction of DHX16 with unanchored ubiquitin is reduced, suggesting that RNA present in a complex might be necessary for recruiting ubiquitin chains. And if you do this also in the presence of RIG-I, you can also see that RNA and RIG-I interactions are RNA is important for this interaction between RIG-I and DHX16. Again, suggesting that there is here a complex between DHX16, RIG-I, and ubiquitin chains that are important for signaling. Now we're going back to how we found this. We found this looking at the effects of the potential role of TRIM6 as an E3 ubiquitin ligase that makes unanchored polyubiquitin chains. So we wanted to know if TRIM6 might be the E3 ligase activating DHX16. So we performed an immunoprecipitation assay, and we found interaction between DHX16 and TRIM6. And very nicely, you can see also that DHX16 is a very nuclear protein. As I said, it's involved in splicing, so most of its localization is in the nucleus, although there is a very small proportion of DHX16 that is present in the cytoplasm. TRIM6 is already known, and we have reported previously that it's localized in the cytoplasm, forming some like cytoplasmic dots in the cytoplasm. But when you overexpress both of them together, you can see that DHX16 mostly relocalizes to the cytoplasm and localizes very, very nicely with TRIM6, again, implicating TRIM6 interactions with DHX16 in some activation of this pathway. We could also show that using TRIM6 knockout cells that these cells DHX16 has a reduced interaction with ubiquitin chains in TRIM6 knockout cells, and also DHX16, as I said, can induce interferon when you overexpress it in cells. But if you do this in TRIM6 knockout cells, there is reduced induction of interferon in these conditions. Again, indicating that DHX16 requires TRIM6 for full activation or induction of interferon response. We also generated TRIM6 knockout mice, and we can also show, like this would suggest that TRIM6 and DHX16 are both implicated in induction of interferon and in an antiviral response. So you can see here that indeed TRIM6 acts as an antiviral factor against SARS-CoV-2. So TRIM6 knockout cells have increased virus replication in the lung. And what is intriguing is that these mice, even though they have higher titers in the lung for SARS-CoV-2, they actually do better during SARS-CoV-2 infection. They appear to lose a little bit less weight loss, and they have a little bit better clinical outcomes. And we're still investigating what the reason for this is. Is probably TRIM6 has other effects in immunological effects that are involved in inflammation that may be affecting pathology. So now really getting into how the mechanistic, like detailed mechanism of how the HX16 might be involved in recognition. We went back to first test how the HX16 might be recognizing influenza virus, because again, as I explained, the HX16 was known to be a splicing factor, and we had evidence that the HX16 might be recognizing or be involved in splicing during virus infection, especially for viruses that splice. So influenza viruses contain two genes, um, the M and NS segments can be spliced to give either M1 and M2 or NS1 and NS2. So we tested by overexpressing the segment 7, which splices to give M1, M2, to see if it would, by RNA immunoprecipitation, if it interacts with DHX16. And what it was really nice is that you can see that there is a weak interaction of DHX16 alone with this fragment with the segment 7 of influenza, but really you have a very good interaction when you have both together, DHX16 and RIG-I. And if you use a mutant of DHX16 that cannot bind RNA very well, you see a huge reduction in interaction with this segment of viral RNA, of the influenza viral RNA. This correlates very well with induction of interferon and ISGs when you have both RIG-I and DHX16 present, but not when you have viral reduction when you have these mutants of DHX16. But what does it have to do with SARS? I mean, SARS does not splice. SARS, it's known to give a genome that it doesn't undergo splicing. So we were curious to know, like, if it would be recognizing SARS, and in a similar assay, in which, but this is with virus infection, in which we overexpress the HX16, you can see that the HX16 pull down can reach RNA that belongs to a part of this SARS-CoV-2 RNA, which is the ORF1 AB segments, and it does not recognize very well, again, when you use the mutant of the HX16. So this suggests that there might be other modes by which the HX16 recognizes viral infection, especially for SARS-CoV-2. It could be that there is a structure in the RNA. To summarize this part of the talk, we identify a novel pattern recognition receptor, DHX16, which can recognize different RNAs of different viruses, including SARS-CoV-2. We're still trying to investigate the detailed pump that might be involved in the DHX16 recognition of SARS. But basically, we have shown that TRIM6 can form unanchored K48 ubiquitin chains that can promote the interaction between DHX16 and RIG-I that help trigger after recognition of the SARS RNA, trigger interferon induction. What I didn't show, and what we think is happening, is that DHX16 is responsible for bringing the proper RNA close to RIG-I to be able to trigger this response. So finally, just as an overall view of what we have been doing, is that we have been working with different E3 ubiquitin ligases that can either covalent or non-covalent modify proteins with ubiquitin, and these interactions with ubiquitin can result either in functions that can be proviral or antiviral, depending on the context of infection, and we have been able to show that TRIM6 can act as an antiviral factor for SARS-CoV-2 via the interferon response, and that the DHX16 can recognize viral infection. With this, I'm going to finish and just thank the people that have been involved. My lab has recently moved from UTMB to Rutgers, where we're starting a new virology center, and thank the main people that were involved in this project, which are Adam Haig and Maria Gonzalez Orozco, and this was not possible without a collaboration with the Pei-Yong Shi Lab and the COVID group, Robin Stevens, Venom Menasheri, Alexander Freiberg, and Janice Ensley at UTMB, and the funding. Thank you all.